presented by Ioannis Michalopoulos. And the rest members of the research team are Georgia Saxami, Apostolos Malapras, Antonia Angelopoulou, Yen. Okay. William Daddy, David Westhead, and Ioannis Michalopoulos. Okay. We're ready? Yes? You may start.
So, okay. Good afternoon, everyone. You might have heard already, but I am Ioannis Michalopoulos, and today I will be presenting the ACT web tool, which is the short name for Arabidopsis Coexpression tool, which was developed at the BRFAA facility under the supervision of Dr. Ioannis Michalopoulos. So, to begin, what was our aim, what we were thinking when we took over this project? So, first of all, we wanted to create, not first of all, the end result was to create a web tool based on a large-scale Arabidopsis thaliana, a model plant, gene co-expression analysis, and through that analysis, the web tool will be able to discover functional partners to a gene of interest and to propose biological roles to genes of unknown function. We could distinguish our project to different steps. The first one being the collection of microarray data and the quality control of it. The co-expression analysis itself, of course. The integration of biological data from multiple external resources. That would be really useful during our enrichment analysis later on. And of course, the creation of the website itself. So, to talk a bit about the scientific basis of co-expression analysis, scientists and researchers all over the world do microarray experiments. And for the purpose of economy, and not in order to not have repeats in experiments, they can deposit their data, both primary and secondary, to public repositories. So they can be freely accessed by everyone in the world. And if someone would be able to filter out and get the best representative samples out of the microarray data from those repositories, they would have a lot of stuff, a lot of data sets to perform a co-expression analysis. The main thinking behind the creation of ACT was that co-expressed genes tend to participate in the same biological processes and in the same biological pathways. So let's say we find a gene that is co-expressed in that specific subset of genes, and we don't know its function, then we can say with a safe measure that it would be part of the same metabolic pathways or same biological processes. So let's start with the first part, the collection of microarray data. First of all, we search for Arabidopsis thaliana as a keyword in the following 3 public repositories. The first one being ArrayExpress, the second one being Gene Expression Omnibus, GEO, and the third one being a specialized Arabidopsis thaliana database of our microarray experiments, which is NASCARrays of Nottingham. And after our search, we found out that more than 50% of the available data, microarray data of Arabidopsis, were conducted on the ADH1 Affymetrix genome array chip. So, our thought was to download all of that data, which whose aliases for platform code was AFE2 in ArrayExpress, GPL 188 in GEO, and in the case of NASCARrays, since it was already offline by the time we had to do the download. We download the whole database which was stored somewhere, and we filtered out the microarray experiments that corresponded to the ADH1 genome array. After that, we found out the corrupted and duplicate experiments, not only from between those three databases, but amongst each database itself. And after the corrupted and duplicate data removal, we ended up with about 1,400 studies and around 20,000 unique samples. So, for the part of those were actually the primary raw data that we got. So, the next step was naturally to convert them to secondary data, which means normalization. We chose the novel scan algorithm, which processes a single array at a time, and so the output results of the expression values are unaffected by any of other samples that you have. So, since I forgot to mention that ADH1 genome array is a single channel microarray chip, so it would be perfect for our kind of Coexpression analysis, which, by the way, also contains a lot of multiple different samples. And the next thing that we did along with the scan algorithm was to use the custom brain array chip description file, version 23, which, in contrast to the older Affymetrix default chip description files, is constantly updated. There's usually 1 or 2 new versions every year. And it does this thing where it it says that a single gene corresponds to a single process, and vice versa, a single process corresponds to a single gene. So we got the guarantee of that one-to-one relation amongst genes and process. And in the end, we got more than 21,000 non-obsolete gene expression values. For our quality control, we used the bioconductor suite in R, and those 3 packages, simple Affy, AffyBLM, and AffyQC report. And after that initial quality control, we removed all the low quality samples determined by the output of those 3 programs, those 3 packages. And after that, we decided to do a secondary quality control by manual metadata examination. And after our metadata examination, we decided to remove all the whole plant and organism samples and all the mutated samples. And we ended up with a total of around 7,400 samples. In order to be able to form a correlation, to discover the correlation between the different genes, we chose the Pearson correlation coefficient way, and I'm going to call it PCC from now on, which is determined by this mathematical formula. And we chose the cause implementation. That implementation says that we can imagine that the 2 variables that we want to correlate with the PCC are like 2 vectors in a 2-dimensional subspace. So we can think of them like this. If the genes are totally co-expressed, then that means that they do this thing. So they put together. No, this thing, I'm sorry. And if they don't have any correlation between them, they are on a 90-degree scale. And if they are totally anticorrelated, they are on -1, so that's like this. And we also chose the Argos implementation because, as you can see in this graph here, compared to the normal distance matrix, the normal distant approach, I'm sorry, for the PCC. It helps us differentiate the extreme values around the edges. So that means that co-expressed and anti-correlated genes would be more distinguished. So you have to bear with me on this one because I'm going to explain all the steps of the next steps of our analysis. So first of all, we got our gene expressions, and using the PCC, we calculated the PCC of each pair of samples by the gene co-expressions, using the gene co-expressions as input, and all of that gave us a distance matrix. The next thing was to use a hierarchical clustering method, which shows the UPGMA, in order to create a sample co-expression tree in nuke format. Then we decided to prove that tree using our own developed in-house file pruning algorithm. And to about 3,500 representative samples. We decided to do that pruning to select the best representative samples of each tissue in an attempt to remove tissue bias, and because we wanted to study the global co-expression landscape of Arabidopsis. After that, the method mirrors those three arrows, those three tables right here. We calculate the PSSA again, but this time we are calculating the gene pair PCC. We get our distance matrix again. Again, we do the UPGMA method, and we end up with a gene co-expression tree in nuke format, and that would also mean the end of our co-expression analysis. Now, up to the development of the website. We created a MySQL database, a functional relation MySQL database, to store the gene expression data. Our experiment and sample metadata, which we also utilized in order to cut the mutated and whole plant samples. And of course, the gene described terms, which will be useful for our enrichment analysis. You might be surprised, but we chose the PHP programming language for all the scripts that we used and created ourselves, and also PHP is the one that, never mind. Also PHP is the one that is going to edit the gene co-expression tree, as you will see later on. Also PHP is the mediator between our database and the website, which is hosted on an Apache 2 server. For the front-end development, we used HTML with CSS, but the Bootstrap, a modern beautification library, and of course, the most precious JavaScript for some specific functions, let's say, for example, the auto-completion of the search button, of the search field. Here I am proud to present you our website in its complete form, hosted in this URL, and this is the main page of our website. Here is the search field, which the user can use to enter a gene of interest. And we also got a few examples right here, which I'm going to use right now to present you our results. So the RS27A protein is a gene codes a ribosomal subunit protein. So if we input RS27A in the search field, the first thing we see after we press the search button is the co-expression subtree with all those genes being co-expressed with our gene of interest. We deem that gene of interest the driver gene, and it's also color-coded yellow. Now, each leaf of the tree is also a single gene, and in this case, we have distinguished between the process ID, and the common gene name of the gene when that is available. If the user clicks on the process ID, it will use that gene as input to the ACT website and produce the Coexpression tree. If the user clicks the common gene name, it will give an outsourced link to Airport, which, in my opinion, is one of the best databases for describing Arabidopsis genes. And finally, here, the user can select an enrichment analysis to perform. There are quite a few enrichment analysis options the user can select. The first one being gene ontology, the second one plant ontology with data got from Platinum, the third one biological pathways from Keck, Wikipathways, and Arachic. And one really important thing is that we also got experimentally verified replacement factor targets from ATG-RegNet. And last but not least, we also got protein domains from BFAM. The overrepresentation analysis on enrichment, we accept both terms here, is done with the hypergeometric distribution, where n is the total number of genes in our database, k is the number of genes that are described by a partitional term in that population, n is the number of the co-expressed genes, or in this case, the number of leaps in the subtree, and k is the term of co-expressed genes that is described by a particular term, let's say ribosomal proteins. And I wouldn't want to spoil the results, but I'm going to do so anyway. So here are the gene ontology aspects for that specific gene we mentioned earlier, S27A. You can see right here that most of the top ranking terms are, of course, related to ribosomes. Molecular function is structural constituent of ribosome. Biological process is translation elongation. And cellular component, everything in the cellular component category is related to ribosomes. And I would like to conclude showing the best aspects, the best advantages of ACT, which is a really simple design, so it can be usable by everyone, and especially by plant molecular biologists, which we also like for them to experimentally verify hypotheses of gene functional gene partnership in biological pathways through the results of ACT. Of course, the fact that we did a really thorough quality control, and from a total of 20,000 samples, we decided to use 3,500 distinct tissue, wild-type representative samples. The modern normalization algorithm of SCAN and the custom brain array CDF. And of course, the fact that we show the co-expression subtree, and the user can also play with it, increase its nodes, decrease its nodes, and also it is really interactive. I would like to thank my supervisor, Dr. Ioannis Michalopoulos, for bringing this project to fruition, and also Dr. Patras and Dr. Ioannis Michalopoulos for actively helping making this project a reality. And of course, Hellenic Greece for sponsoring us in order to make this happen. And also all of you who listened to this presentation. Thank you.
Thank you very much for the very, very nice talk. Are there any questions? Yes, please.
Congratulations. If I understand it well, this is an old tool that it's going to be updated now. Is it true? And not
yes.
The question is.
The fact excuse me for a moment, we changed so much in the analysis that it's considered a total different version of it. Yes.
The question is whether it will be possible to to have this pipeline as a tool for a general purpose, not only for Arabidopsis data,
Yes.
but would it be possible to have a tool that a user can submit a number of
I suppose that all the scripts that we've written can be translated to a pipeline, although that would honestly take quite a bit of work. But other than that, I think, all the algorithm, the UBGM algorithm perhaps is done through an R package. The PSS calculated using PHP scripts. The pruning algorithm is also through a PHP script. I think all of this can be put together with some effort.
Thank you very much. That will be my question too. Yes.
Two questions. How do you treat anti-correlated genes? And second question is that when you combine microarray data,
Yep.
you assume that there is a global expression pattern for each gene. How would you comment
Okay,
on
so
this?
let's start on the first thing. Unfortunately, the thing with our kind of expression analysis is, although we have a distant matrix with the anti-correlation values of 180 degrees, 2, and the co-expressed values of 0 degrees, during our hierarchical clustering, the results of the two really all take only into account the co-expression aspect of that analysis. And excuse me, repeat the second question again.
The second question is that when you combine microarray data sets, you assume that each gene has a global expression pattern. Is that true? How would you comment? For example, if you
Yes.
take half of these data sets, the expression pattern will be different. So
I think the scan algorithm is what makes the deal for us because it uses each sample differently. And in the end, whatever data we get out, we just accept it as it is. I think that the most important thing is actually picking out the representative samples and not the total amount of data that we start with.
Okay, thank you very much. Another question? Okay, let's thank again Mrs. Michalopoulos. We have reached the end of the session much faster than expected due to the punctuality of the